In the face of climate change and growing drought concerns, there is new research that is shining a light on the immune response in plants that could be key to harvesting drought-resistant crops in the future.
And if we can understand the function of these genes in this reference plant, we can then translate that information to a variety of crop plants.
Salk scientists, including Dr. Joseph Eckert, examined this flowering plant commonly used in plant research. You can see these leaves are dried up, discolored, and showing signs of drought stress.
So these are actually the plants we're interested in here because when we've mutated the gene, it can't recover anymore.
Studies on how plants respond to drought conditions are well known, but their recovery has been understudied. With a focus on drought recovery, the quick response from plants when water is reintroduced and the reaction itself was astonishing to researchers.
So that was a surprise to us that the genes that are involved in the recovery process are very rapidly induced. Within 15 minutes, thousands of genes are increased, and they're not the same genes that are going down during the drought process itself. It's a completely new set of genes.
It's not just that the plants were thirsty. When absorbing water, a plant's main priority was to boost its own immune system. This plant gene response is believed to prevent infection from harmful bacteria during that recovery period.
So they're turning on pathways that we actually began to test, and sure enough, those plants are activating genes to protect itself.
To see this reaction requires a deeper look at the plant leaf, or section of that leaf, using advanced gene sequencing and imaging technology in which you can see the genes light up.
That diversity, yeah, and in different conditions, it does something completely different. And these are like the first times we can really see expression of a gene within the actual location in the leaf.
Researchers, including Netanella, refer to the immune response as drought recovery-induced immunity. From the lab to the field, this gene research highlights a broad concept that can be applied to various different plants.
And if we can really understand how plants are recovering for that mild drought, maybe we can really understand when you're pushing them too far, how we can make them more resilient.
This same immune response also took place in wild and domesticated tomato plants. Additionally, many of the genes that make up the lab plants are shared in various plant species, including agricultural crops like wheat and rice.
We will look into gene expression in every cell type of the leaf tissue, of the crown tissue, and of the root tissue. So we will have a very comprehensive understanding of how each gene is expressed during drought and recovery.
This is sorghum. It's an important multi-purpose cereal crop used globally for flour and baking goods, sweeteners, and grain bowls. It is also drought tolerant, so its gene response will help expand this research.
If we understand the recovery process, it may help us help the plants to recover better.
And keep our food supply growing as the climate continues to change. For CBS 8, I'm Chief Meteorologist Carlene Chavis.